Hello, student. This is Dr. Faizan Mirza. In this video, I'll be discussing with you the questions that I've taken from the chapter of energy and respiration. So this question, October, November 2021, and variant 41. They gave this whole sequence of events here in which you can see the first three stages of respiration are shown to you. They have shown to you how glucose is converted to triphosphate and then pyruvate. We know this is glycolysis. After glycolysis, we can see that they have given to us the link reaction as well. And from link reaction, they have given us Krebs cycle. They have already told you here that these steps are occurring twice because we know that once pyruvate is produced, we know that link reaction and Krebs cycle occur twice for each glucose molecule. So the ATP is being consumed, they are shown, and the ATP is being produced, they are shown. The reduced NADs, reduced FADs are also mentioned. So all the steps are shown here. What are they asking? Number one, they're asking name the molecule Y in figure 6.1. So the molecule Y, we locate Y, this is Y here. So pyruvate, something is being removed from pyruvate, and these are two molecules being removed. What should these be? Because already they have mentioned that two reduced NADs are being removed. We know that two reduced NAD and two carbon dioxide molecules are removed from pyruvate, so it becomes like a stalcoate. So this should be carbon dioxide. Later they're asking how can we know from figure 6.1 that glycolysis involves oxidation? We know oxidation means that something must be getting reduced. So reduced NAD is being produced. So yes, that will get you a mark. Plus, we can see that the transphosphate is being produced. So that actually shows to us that yes, uh, this is uh, this is an oxidative pathway. Next, the question where they are asking us, at one time it was thought that oxidative phosphorylation would yield one reduced NAD and one reduced FAD actually would give you three ATP and two ATP molecule respectively. Using figure 6.1, they're asking us to calculate the theoretical value for the net number of ATP molecules that are synthesized for each glucose molecules, and we have to show the calculation. Then they are saying modern research has shown the actual net number of ATP molecules synthesized in glucose respired is much lower than theoretical value. They are asking to us using figure 6.1, calculate the theoretical value for the net number of ATP molecules that are synthesized for each molecule of glucose respired in all phosphorylation reactions. So the wordings are very important here. We need to take into consideration all phosphorylation reactions for each molecule of glucose. So let's go back here. When we know that when glycolysis is occurring, so glycolysis ends up, it actually uses first two ATPs and there's a net gain of four ATPs. So we know that four ATPs are produced and two ATPs are being used up. So yeah, the net gain is two ATP from glycolysis. We move to Krebs cycle. We know in this in your link reaction, we know that link reaction only produces reduced NADs, but ATP is not produced there. Let's move to Krebs cycle. Krebs cycle ends up producing two ATPs. They have mentioned to us this as well. Now, how many reduced NADs are being produced? If you look at this figure, so let's start counting the number of reduced NADs they have shown to us. They are showing two reduced NAD are produced here, then two reduced NAD are produced here. We add two reduced NADs, and then there are six reduced NADs which are produced in Krebs cycle. So a total of 10 reduced NADs are produced overall in these three stages. We know from the theoretical value that 3 ATP are being produced by each reduced NAD. So a total of 10 reduced NAD are being produced. So they will end up producing 30 ATPs. Coming to reduced FAD, there are two reduced FAD being produced. We are told that the two ATPs can be produced per reduced FAD. So we multiply 2 by 2, we get 4 ATPs. We add all these values together, 2 plus 30 plus 4, we get 38 ATPs. So this is the answer. Although they have given us very little space there, but yeah, you can just mention which reactions you are using to calculate the net gain of ATP. You have to take into consideration all the phosphorylation reactions and this ratio which they have provided to us for the theoretical value. Next. So there's two reasons why the actual net number of ATP molecules synthesized is less than the theoretical number. We know that ATP's energy is being used in moving the products of glycolysis, such as pyruvate and reduced NAD, into the mitochondrial matrix from cytoplasm. So that will consume ATPs and energy. Some energy is lost as heat. Plus, products of glycolysis may enter some other metabolic pathway within the cytoplasm. Outline the roles of NAD and FAD in respiration. We can mention the reference to coenzymes and how they can carry hydrogens from cytoplasm to the matrix and then to cristae eventually. They get reduced in glycolysis, link reaction, Krebs cycle. They participate in dehydrogenation reaction during electron transport chain and oxidative phosphorylation, as well as when they are being produced. So yeah, you can add those points and get 3 marks. They're just asking you to outline here. You don't have to give the detail or explain anything. Next, they're asking us to us. They are asking us that rotenone is used as an insecticide and it kills by inhibiting the transfer of electrons in the mitochondria. So electron transport is being affected. Explain how rotenone affects ATP synthesis in mitochondria. So if ETC is obviously if electron transport chain is being compromised, so electron transport chain is not getting completed, electrons will not fall into matrix eventually. Plus we can expect that there will be a lesser quantity of all these things. Where there will be a reduced chemiosmosis occurring. There will be less proton gradient being established. We know that a lower number of protons will start to move from ATP synthase through ATP synthase, because since they're less ATP, less H plus gradient is being there, so less H plus will move into the matrix, and then eventually less ATP will be produced. So I hope this question becomes easy for you to comprehend and to attempt. Let's move on. Then this is the other variant, October, November 21, again the same year, but the other variant for two. In this, what they give, initially they're asking to us just the general information of describe the properties of ATP, how it is suitable for a universal currency. Because this question comes more often. This question came a lot of times earlier as well. Some cramped facts that you need to state. ATP is water soluble. ATP is a small molecule. This allows it to be moved freely along the cytoplasm and the matrix. ATP can participate in hydrolysis reaction. Removal of a single phosphate group from ATP will produce 30.5 kilojoule energy. And we can add that ADP and PI can rejoin to produce ATP using energy from substrate level reaction and oxidative phosphorylation. Yeah, all these can be added. You just have to give a description here. Suggests why ATP is needed for protein synthesis. So here they're expecting you to recall your AS level. So we know that in protein synthesis, the nucleotide need to be activated so that they can be added into mRNA. Amino acid joining also requires ATP. Moving the ribosome along the mRNA would need energy as well, because that's a movement. Wherever movement is occurring, ATP energy is being used. Wherever in the cell, whichever process is occurring. Figure 6.1 shows you a diagram. There are regions given as A, B, C, D, and E. Here they have shown to you each membrane of the mitochondria as a double layer. So the outer membrane is shown as 2 layers, and the inner membrane is also shown as 2 layers. So they have shown you 1, 2, 3, and 4 layers here, because they are separating each phospholipid layer separately as a line. This is the outer membrane, one phospholipid layer, and then the other. This makes the whole outer membrane. And then the inner membrane, which is folded again, is a phospholipid bilayer. So they have shown to you it has 2 lines. So the length is provided as 5.25 micrometer. The diameter is 0.72 micrometer. Now, using these values, what they're asking us to complete the table using letter A to E. Each letter may be used once, more than once, or not at all. Where will the Krebs cycle be occurring? So it's occurring at A. Why it's occurring at A? Because A is the region which is the matrix. So yeah, it will be occurring at A. Then phospholipid bilayer for impermeable to H plus ion, where H plus ion cannot move. The answer could either be E or D. So we know that this region is impermeable to H plus ion. Why is it impermeable? Because we know that protons move from this membrane and not the other one. So, and plus we can use this the other one mentioned is D as well. Site of translation. Translation we know occur on ribosomes. So the structures shown to us, these C structures, they must be the ribosomes. That's why they are shown to us separately. So this is C. Assume that the mitochondria is a cylinder. Calculate the surface area of mitochondria using the formula of surface area of a cylinder. They have given to us formula 2 pi r square h plus 2 pi r h. So you need to look at the diagram again to see what's the radius and what's the height of this particular mitochondrion. So what have we done? We'll just multiply these accordingly. The values provided to us are 0.72 diameter and 5.25 length. So 5.25 length becomes the height. And we know the diameter. The diameter provided to us is 0.72. We can divide this by 2. And when we divide this by 2, we will get the value of the radius. So the answer of 0.72 divided by 2 will be our radius. So we put the value here. This is 0.36, which came by dividing the diameter by 2. So that's the radius. So we just square the value of radius and then multiply it eventually and get the answer 0. In the space provided in micrometer square. Towards the end, they're asking the inner membrane of mitochondria has much larger surface area than the outer membrane because of the presence of cristae. Different cell types may have varying number of cristae in the mitochondria. Cardiac muscle have mitochondria with very large number of cristae. Suggestion explain why cardiac muscle cells would have these. So yeah, more cristae would mean more electron transport chain occurring, more chemiosmosis, more ATP being produced, a lot of oxidative phosphorylation occurring. Cardiac muscles need to contract and relax continuously, so a very large cristae exists, so that actually will just assist the functioning or the physiology of these cardiac muscle cells. So I hope this video was helpful for you. I would prefer if you attempt these both questions yourself and to see and try to see how well you understood the working. That's all from my side. Thank you so much.